Okay, our presentation is on transduction, and I'm Luke.
And I'm Scott. Okay, so introducing to genetic secrets. How does bacteria share their survival strategies? Well, bacteria need a way to adapt quickly and especially against threats like antibiotics. So they would do a genetic recombination, gives bacteria new traits like ability to survive antibiotics or other harsh conditions. Even though bacteria can reproduce asexually, these mechanisms would allow them to trade DNA like tiny survival blueprints. And this would make bacteria population evolve much faster than other organisms. And the three mechanisms for horizontal gene transfer is conjugation, transformation, and transduction. And we will be going over transduction.
Okay, so what is transduction? Transduction is the process where a bacteriophage or virus transfers DNA from one bacterial cell to another after the original cell it infected dies. This allows bacteria to share genes, including those for antibiotic resistance, without direct contact between the bacterial cells. So an example, or this would be like the bacteriophage would be a delivery truck for DNA, and then when it infects the bacteriums, it picks up pieces of that cell's DNA and then it carries it to the next bacteria it infects. This process doesn't require the two bacteria to touch, and the virus does all the transferring between them. That's why transduction is such an efficient way for antibiotics resistance to spread.
Okay, the step-by-step process. So first, the phage would inject its DNA into the bacteria, and the phage enzyme would slowly degrade the host DNA, and it would slowly kill off the bacteria actually. And as the cell synthesizes new phages that incorporates the phage DNA, and it would mistakenly synthesize the some of the host DNA. And this would lead to transducing the phage injects into the donor's DNA. And the donor's DNA is incorporated into the recipient's chromosome by recombination. And the key part of the of this process is the phage can accidentally act as a genetic messenger, mixing the bacterial DNA between different cells.
So there's two types of transduction, and these are generalized transduction and specialized transduction. In generalized transduction, it happens when a bacteriophage accidentally packages random fragments of the bacteria DNA instead of its own viral DNA during the Leydig cycle. Specialized transduction is when lysogenic bacteriophages remove itself from the bacteria chromosomes incorrectly, and this makes it take specific genes near the viral integration site along with it. This makes it more predictable for scientists can identify which bacterial genes are likely to be moved. Specialized transduction can also spread dangerous genes, such as those that produce bacterial toxins.
Okay, so why does transduction matter? Well, it matters because it spreads beneficial or resistant genes quickly into bacteria population. And because of this, it could create uh, bacteria that are antibiotics resistant, making them more of a superbug. And this would play a major role in horizontal gene transfer, helping bacteria evolve much quickly. And this image at the bottom, it shows how lysogenic a phage can carry antibiotic resistance genes and spreads them between bacteria. So first, the virus inserts the resistance gene into the bacterium, and the cell now would acquire resistance. And from there, the gene was spread into two different ways. Vertically, which is offsprings, and horizontally, which means the phage transfers the genes to other bacteria. And over time, this allows resistance to spread not just into one specific type, but across many different types of species of bacteria. So this antibiotic resistance can move from other bacteria to another species, making an environment bacteria that's harmless into a dangerous that could have a human cause a human illness.
So, compare transduction with other gene transformation transfer methods. Uh, Conjugation and transformation are the two other ways. And conjugation works through transferring pilus. or a bridge-like structure and having that go in between the two bacterial cells, and then it transfers the DNA through that. This does require direct contact, or at least for the bacterial cells to be extremely close to each other. An example of this would be E. coli F plus and F minus cells. In transformation, there's an uptake of naked DNA from the environment, and this does not require contact because no other bacterial cells need to be nearby when it does this. An example of this is the Griffiths experiment. Transduction our way, like we explained earlier, has gene transfer through bacteriophages, and it does not require contact. An example of this would be the lambdaphage. So, in conclusion, transduction uses bacteriophages to transfer bacterial DNA. It creates genetic variation and can spread antibiotic resistance. It's one of the three main DNA exchange methods, with conjugation and transformation being the other two, and it's a cause for many public health concerns and makes many bacterial cells resistant to antibiotics. Transduction is also more than a biology term, and it's a real-world problem. It explains how bacteria adapts faster than we can develop new antibiotics, and understanding this helps scientists find better ways to control infections and design smarter treatments.
Okay, so these are the site pages, and that's it. Thank you.